Today we're here at Vanderbilt University in Nashville, Tennessee, and we're very lucky to have been invited to interact with many of the scientists and clinicians that power this type 1 diabetes program here, which is one of the best in the country. And we're very interested in talking with scientists and having them share and showcase their work. And our very first discussion here is with Dr. Danielle Dean. We're very happy to have her here again. We spoke with her 2 years ago, and she, if you don't know her, she's an assistant professor in medicine at Division of Diabetes, Endocrinology, and Metabolism in the Department of Medicine at Vanderbilt University Medical Center. And her interests are how nutritional status and environmental gene interactions contribute to aging and diseases like diabetes. While much effort has been focused on understanding beta cell biology because of insulin's well-known role in diabetes, very little is known about signals regulating other islet cells, such as alpha cells. Alpha cells secrete glucagon in response to hypoglycemia, but persons with diabetes have hypergluconemia contributing to hyperglycemia. And in an effort to understand the molecular mechanisms that determine alpha cell mass in the pancreatic islet, Dr. Dean identified a liver alpha cell axis, a crosstalk between liver glucagon signaling and alpha sensing of amino acids. She started her independent laboratory at Vanderbilt in 2019, and much of the lab's focus is to understand how a liver alpha cell axis regulates islet cell growth and how this axis may contribute to liver and alpha cell dysfunction in obesity and diabetes. Today, she will comment a little bit and extend that interest and her discussion further into type 1 diabetes. Thank you so much for joining us, Danielle, and we're really happy to have you.
Thank you so much. I appreciate you inviting me to come tell you about our lab's work. Really, I think we're doing some really exciting work, especially on identifying what makes an alpha cell and what are the real drivers of alpha cell function and alpha cell mass. And, we think that we can use some of this information to help us figure out how to treat patients with type 1 diabetes better. And so can we target the alpha cell to either restore glucagon signaling in hypoglycemia, or can we even use the alpha cell as a target to restore beta cell mass and maybe have a little different way to cure or treat diabetes. So my learning objectives for you today are to talk about what regulates alpha cell mass and function and ask, are they the same? I'm going to talk about what the liver alpha cell axis is and how islet cells sense amino acids. And then I'm going to also talk about how alpha cells and beta cells may sense amino acids differently. And so, they may not do this the same way. So I posed this question, what makes an alpha cell an alpha cell? And I think that what you can gain from that is to try to think about, what do we know about alpha cells, and can that give us some insight into what the real function for these cells are? And these are not my data. These are from Pedro Herrera's group, where they took bulk sorted RNA sequencing and looked at, in human cells, what are the top markers expressed on individual endocrine cell types. And so here on the top right, you can see the beta cell, the top markers being things like insulin, of course, and IAPP, the amylin protein that's co-secreted from beta cells, but also things like G6PC2, things that regulate glucagon catabolism, glucose breakdown in beta cells. So I think that gives us a clue as to the sort of signals that regulate beta, or the sort of things that make a beta cell a beta cell. But on the other hand, if you look at the alpha cell, we don't see things related to glucose metabolism. And that is interesting because glucagon, the word, is a contraction of glucose agonist, having to do with how it was discovered over 100 years ago now. And but what you really see is, of course, things like glucagon that are the top markers of alpha cells, but also these genes like GLS, which stands for glutaminase. This is the rate limiting step in glutamine catabolism. So converts glutamine to glutamate. And then genes like SLC7A2, which are catabolic or cationic amino acid transporters. And so this transporter transports arginine, lysine, and ornithine. Of course, we know that arginine is a potent stimulator of glucagon secretion. So I think that this gives us a clue that maybe the role for an alpha cell is really to sense amino acid tone. And so we're going to dive a little deeper into that. Now, I will qualify. I'm not saying that glucose isn't important for an alpha cell. I'm just hoping that you broaden that thought into other nutrients that might regulate alpha cells. So what happens? So what do we know about glucagon? So what happens if you interrupt glucagon signaling? We know if we give glucagon, that can rescue hypoglycemia in severe cases. And if you block glucagon signaling, you kind of have the opposite effect where, giving glucagon can restore glucose levels when they're very low. And then if you block glucagon signaling, you can actually lower blood glucose levels. So there's this very clear relationship between glucagon and glucose, which is why it got its name. And so you can see this here, especially in these mice, this glucagon receptor knockout mice. This is a global knockout. And you can see that fasting glycemia is improved in these mice as well as their glucose tolerance. And if you use a small molecule to also block the glucagon receptor, you also get improvements in fasting glycemia. And so this seems like this might be something that could be useful to treat diabetes. But the problem with these drugs is that they also cause severe hyperglucagonemia. And so you can see that here where the control animals versus the antibody treated against the glucagon receptor, those antibody treated mice, they have severe fasting hyperglucagonemia. And this is partially due to an expansion of the alpha cell mass. So here on the left, you can see a wild type mouse's pancreas, amylase marking the exocrine tissue, the acinar cells in red, and then the green marking these alpha cells and staining for glucagon. You can see that glucagon mass is very low in a normal pancreas, but in these glucagon receptor knockout mouse, the alpha cell mass is increased by 13-fold by 22 weeks of age. So they develop this massive alpha cell hyperplasia that eventually turns into tumors as these mice age. So the alpha cells continue to grow, and you can even detect tumors. And it turns out that if you block glucagon signaling by any mechanism in liver, this leads to alpha cell proliferation and hyperplasia. So this shows that there's this very strong relationship between liver signaling of glucagon and this compensatory or feedback to the alpha cell that tells it to grow. This happens in humans as well. So here I'm showing data that if you take human islets and transplant them into immunodeficient mice, treat them with the same glucagon receptor monoclonal antibody that I showed you earlier, and then look at their glucagon content in those human islets, as well as their alpha cell proliferation as measured by Ki-67 staining, you can see that there's an increase in both glucagon content after 4 weeks, as well as an increase in alpha cell proliferation by 2 weeks of treatment. And this mirrors the disease that we see in humans called Mavash disease, which is due to biallelic mutations in the glucagon receptor. And you can see that these patients develop these glucagon microadenomas. This is the glucagon staining here in green. And so this shows you that human alpha cells, which is really important, human alpha cells can also respond to whatever mitogenic stimulus is stimulating these cells to grow when you interrupt glucagon signaling. And the reason that's so impactful is because we know that human beta cells are much more resistant to growing. Human adult, these are adult islets, they're much more resistant to growing in response to any stimuli. So we worked really hard to try to identify what were the specific factors that stimulated alpha cell growth. And we looked for a lot of secreted factors, little proteins that were changed. And the way that we did this is by screening serum for proteomics from the mice that had interrupted glucagon signaling. We did serum metabolomics. We also did liver transcriptomics from the liver of, as you can see here, glucagon receptor knockout mice in red and glucagon receptor monoclonal antibody treated mice in blue. And what we found is that there was this shift towards amino acid transporters as well as amino acid catabolic enzymes being downregulated. And so what these changes in gene expression predicted is that there would be an increase in serum amino acid levels. And that's exactly what we observed. So for almost every amino acid, we saw about a 3 to 6 fold increase in circulating levels of these amino acids in various models of blocking the glucagon signaling. And particularly, we saw 2 amino acids that caught our attention, one being glutamine, which of course is that target of glutaminase. Shown here, you can see there's about a 6-fold increase in circulating glutamine levels in mice that have interrupted glucagon signaling, as well as arginine. They have about a 5 to 6-fold increase in circulating levels. So this led to this idea that there's this liver alpha cell axis where glucagon communicates to liver and amino acids could feed back possibly to the alpha cell to regulate not just secretion, which we've known for a very long time. This was discovered in the 1960s, but also to regulate alpha cell mass. And so we wanted to test that. And so we created media that mimics the concentrations of amino acids that we measured empirically in the serum of mice. So here, low amino acid means wild type levels of amino acids in blood, and high amino acids means glucagon receptor knockout serum levels in the blood. And you can see that you see a nice stimulation of alpha cell proliferation when we take islets from mice and culture them for 3 days in these culture conditions. And we know that this is glutamine dependent because we can keep all other amino acids high and only change the concentration of glutamine in the media, and we can dose dependently stimulate alpha cell proliferation with increasing amino acid levels as we pass the point where we go from normal circulating levels around 500 micromolar up to this 3 to 4 millimolar levels of glutamine that we observe in the blood. And what's really remarkable is that at least in these short conditions, this is very selective for alpha cells. So glucagon signals to the liver, regulates through the glucagon receptor, not just glycogenolysis, but also gluconeogenesis, as well as ureagenesis. And this is through the fractional extraction of amino acids from blood. And then amino acids, of course, can feed back on the alpha cell and regulate glucagon secretion through stimulating the alpha cell. And if you disrupt glucagon signaling, this leads to an impairment of this amino acid clearance. The alpha cell actually does some very interesting things. It actually increases the number of amino acid transporters that it expresses, and that leads through an mTOR-dependent mechanism. It's rapamycin sensitive, we showed in publications, to glucagon hypersecretion as well as alpha cell proliferation or hyperplasia. And so this is at the heart, the liver alpha cell axis and the changes that occur. So we wanted to understand how this alpha cell hyperplasia was occurring. And one of the things that caught us our attention was this 23-fold increase in SLC38A5 expression. This is a glutamine transporter. And I showed you that glutamine was important for regulating alpha cell proliferation. And so we thought this transporter was going to be the driver of alpha cell proliferation in response to hyperglutaminemia. And so we tested this using a alpha cell specific SLC38A5 knockout model where we inducibly knocked out SLC38A5 expression. Here I'm showing you female mice. We've done this in male mice as well. And after 2 weeks, we started treating them with a glucagon receptor monoclonal antibody. And so since I told you that SLC38A5 is induced, it turns out it's not very highly expressed in adult alpha cells. You have to induce this expression with treatment of the monoclonal antibody or knocking out the glucagon receptor. But you can see that we nicely knocked out this transporter. And surprisingly, we didn't have that much of an impact on proliferation. So you see maybe a little bit of a dip, but it's not significant. It's not significant in the males either. So we think that this transporter may be doing something else regarding regulating alpha cell proliferation. Now, we don't have the whole picture. And it suggests to us that there's probably other amino acids that are playing really important roles and that could even compensate for this transporter being knocked out. So, we knew that glutaminase was highly expressed in alpha cells. And so we wanted to understand, well, okay, we know glutamine is important, but what is the fate of glutamine in these cells? And how is that impacting alpha cell proliferation? And so we thought that because glutaminase was highly expressed in alpha cells, you can see this here using data from our calcium group. You can see that mouse alpha cells express very high levels of of glutaminase as well, and very low levels of the liver isoform of glutaminase too. And so we wanted to understand if we could block, excuse me, and you can see that here in staining as well. Here we've stained a mouse pancreas for glutaminase. You can see that it's expressed throughout the pancreas, but it's much more highly expressed in alpha cells, and seems to be organized into these punctas as well. And what's more than that is you also see expression of this GAC isoform, which is a highly active form of glutaminase that is more typically expressed in cancer cells, actually. And so alpha cells are taking maybe a play from cancer cells books to hijack glutamine transport and catabolism to drive their cell growth, which we find very interesting. What's important, though, to note is that we don't see changes of glutaminase expression in human pancreas. So here I'm showing you non-diabetic human pancreas. We've looked in type 1 as well as type 2, and we see that alpha cells express very high levels of glutaminase, but that's not changing. And we think that's important for thinking about how this pathway could be involved in dysregulation of glucagon secretion in type 1 diabetic alpha cells. So, as I alluded to, we wanted to target this. So first we started with a small molecule inhibitor that's actually been studied for treatment of cancer, this CB839. It reduces the proliferation of cancer cells, and we wanted to see if it could reduce this proliferation of alpha cells in response to this high amino acid media. And of course, what we found is that you can dose-dependently inhibit alpha cell proliferation in our mouse islet culture model with the CB839. We next went on to look at how loss of, these got out of order. We wanted to look at how loss of glutaminase expression in mice, in alpha cells specifically, might impact both glucagon secretion as well as alpha cell proliferation. And so we created this glutaminase specific or this alpha cell specific glutaminase knockout mouse. And you can see we have very nice knockdown of glutaminase expression. And when we looked at glucagon secretion, we were kind of surprised. We really thought that glucagon secretion was going to be impacted by targeting glutaminase expression in alpha cells. And while we've tried to stimulate glucagon secretion with a bolus of glutamine, we actually haven't been able to get enough glutamine in to these mice to stimulate secretion, as well as we haven't been able to really stimulate alpha cell function or glucagon secretion with glutamine in culture either. So we actually are really interested. We know that glutamine is absolutely required for glucagon secretion, but we don't feel like it is a very good secretagogue. And so we went after arginine instead. And so when we used arginine as a secretagogue for glucagon secretion, we actually didn't see much of an effect at all. So we know that glutaminase expression appears to be dispensable for glucagon secretion, but we don't really know anything about alpha cell proliferation. So we went in and we treated these mice with the glucagon receptor monoclonal antibody, and you can see that we get a nice knockdown or a nice loss of Ki67 positive alpha cells in these glucagon receptor monoclonal antibody treated mice. And again, we also impact SLC38A5 expression in these mice too, showing us that we're starting to disrupt these downstream pathways by targeting glutaminase expression. So glutamine metabolism seems to be very important for the alpha cell proliferation and maybe somewhat dispensable for the secretion. So that led us to think about what other amino acids could be regulating both alpha cell proliferation as well as glucagon secretion. And you know, this paper here by John Garrick's group was published 50 years ago, showing that if you perfuse the rat pancreas, not perfuse, perfuse the rat pancreas with arginine, this is under low glucose conditions, you get this beautiful biphasic and dose-dependent stimulation of glucagon secretion by arginine. And this looks a lot like what we would expect to see for glucose and insulin secretion. But if you look at insulin secretion, you see this really sluggish, very slow increase in insulin secretion in response to arginine. And if you change this and you stimulate under high glucose conditions, you see the opposite. You see that beautiful biphasic response to insulin secretion from both the glucose and arginine. And you do see a suppression of glucagon secretion in high glucose, but you still see stimulation because it can't overcome that strong increase in secretion that's due to the arginine stimulation. So this makes us think that alpha cells and beta cells are sensing arginine tone differently. And so we wanted to understand what the role of arginine in glucagon secretion as well as alpha cell proliferation were. And the way we started to look at this is we looked at the transporter expression. And as I alluded to in our first slide, that SLC7A2 is highly expressed in human alpha cells. We see that it's not just highly expressed, it's the most highly expressed amino acid transporter in human alpha cells. And it's about at least 3 to 4-fold higher in alpha cells versus beta cells in human as well as in mice. And so this made us think that this is the major arginine transporter. So we next look to go look and see if arginine stimulates alpha cell proliferation. And the way that we found this is through our original screening to understand which amino acids regulated secretion. We batched amino acids together and lowered them down to wild type amino acid levels to see if we could reduce alpha cell proliferation when we kept all other amino acids high. And we thought we could identify individual amino acids that were indispensable for the alpha cell proliferation that we observed. And one of those was arginine. So here I'm showing you all amino acids high here in purple. And then if you lower this batch of amino acids, glycine, arginine, and histidine, we did other batches too. I'm just showing you this one. But if you lower this one, you get a reduction in alpha cell proliferation. And only when you restore back arginine levels do you restore alpha cell proliferation in response to high amino acids. And again, this is important because we know that this transporter is highly expressed. And so this really supports the idea that arginine transport through this transporter is what's driving alpha cell proliferation. And so we wanted to test that. And so the way we tested that is using SLC7A2 knockout mouse. And this is a global knockout mouse. And you can see when we knock out SLC7A2 here in the white bar and treat those islets with high amino acid media, that we lose alpha cell proliferation in response to the high amino acids. And so this shows that SLC7A2 is indispensable for this alpha cell proliferation. We did a lot of studies in vivo as well in mice, and we see the same thing. All right. So what about secretion? So we looked at arginine stimulated secretion in these knockout mice, and we found that there was no stimulation of glucagon secretion in response to arginine bolus. And if we take those islets out of the mouse and then perfuse them, you see this beautiful low glucose and arginine stimulated secretion as well as KCl in wild type, or these SLC7A2++ mice in blue. But if you look at the islets from knockout mice, there's really no response to any secretagogue, including KCl, in this condition. And there's no change in the alpha cell mass. So this appears to be some sort of intrinsic islet secretory defect from these mice. We see similar effects on arginine-stimulated insulin secretion, and then in vivo. And then if you look at the perfused islets, you can see that they have a modest impact on their glucose-stimulated insulin secretion, but their arginine-stimulated insulin secretion is severely impaired. All right, so this didn't sit well with us because of this difference between expression in alpha cells versus beta cells. And we thought, well, if an alpha cell has such high expression of SLC7A2, then, is the alpha cell maybe sensing the amino acid tone and communicating that to the beta cell? And so we wanted to test this idea to understand, do beta cells really sense arginine tone intrinsically, or does the alpha cell do that for the islet? And so to test this, we made SLC7A2 flox mice and then crossed those mice to either INSCRE ER mice, or we crossed them to the glucagon-CRE ERT2 mice. And throughout the rest of the talk, you'll see green is the flox mice, purple is the beta cell specific, and then the blue are the alpha cell specific knockout. So again, we went back to our in vivo arginine stimulation, and the first thing that we observed is that in the alpha cell specific knockout and not in the other mice, we observed a deficit in arginine stimulated glucagon secretion. But what really caught our attention is that when we looked at insulin secretion in response to arginine, we saw that it was still stimulated in the beta cell specific SLC7A2 knockouts, but the alpha cell specific 7A2 knockouts had a loss of arginine stimulated insulin secretion. And so this suggested to us that these higher levels of amino acid transporters on the alpha cells versus the beta cells suggested that if we knock out the beta cell versus knocking out the alpha cell, we don't see a strong effect on glucagon and insulin secretion if we target the beta cell, but we see a strong effect if we target the alpha cell expression of SLC7A2. So then again, we went and did parafusions on these islets. And so you can appreciate here in green that we have nice glucagon secretion. If we look at the beta cells, they have a pretty good stimulation of glucagon secretion in response to arginine, as well as low glucose. But if we look at the alpha cell specific knockouts, they look a lot like the global knockouts, where they don't have a very strong response to the arginine. But what really caught our attention again is that these alpha cell specific 7A2 knockouts don't have strong sensing of arginine, strong stimulation of insulin secretion in response to arginine. And so this is really supportive of this idea that alpha cells are acting as amino acid sensors for the islet and promoting insulin secretion through proglucagon-derived peptides. And I show you a list of publications on the right side that I think have really championed this idea that the alpha cell and the beta cell are really communicating to each other through paracrine factors to regulate each other's secretion very, very strongly. And most of these studies have targeted glucagon receptors or GLP-1 receptors. And I think what makes our study unique is that we've actually targeted the amino acid sensing mechanisms and shown that the same thing, that there is a deficit or that there's communication rather between the alpha cell and the beta cell to regulate insulin secretion. All right, so that leaves us with the liver alpha cell axis. Hopefully I've convinced you that amino acids stimulate glucagon and they regulate alpha cell proliferation. But I want to leave you to that, fatty acids also can impact the alpha cell. And I think we're seeing some really nice data coming from people like Jacob Knudsen and James Cantley, thinking about how glucose and fatty acids interact to regulate alpha cell function as well. So, it's not just amino acids, but probably the combination of nutrient tone is really regulating these cells. And I couldn't do my job if I said that it was just down to intrinsic nutrient sensing, but there are also endocrine factors from the gut like GIP. They're very important for this and probably are really critical for the amplitude of amino acids stimulating glucagon secretion. And then the last thing I want to leave you with is this idea that is the alpha cell secreting anything else? Like, we know that beta cells secrete amylin and other peptides, but I showed you the glutaminase data, and the end product of glutaminase is glutamate. And glutamate is also an alpha cell product. It's secreted in vesicles, and beautiful data from Alejandro Caseda's group and others have shown that glutamate, restoring glutamate signaling in alpha cells could be a potential therapeutic for alpha cell for for hypoglycemia. So I think there's some awesome opportunities there as well. So these are the folks who did this work. This is the members of our lab, as well as our collaborators over here. And I couldn't do this without the funding that we've been so fortunate to receive as well. Thank you.
Thank you so much, Danielle. That was just an absolutely perfect walkthrough the mechanism of the alpha cell as it's known to date. I wondered, I had a couple questions for you. I really am interested in this whole fact or this whole idea of alpha cells setting a tone basically for the beta cells. But what is your thought about, this whole idea of rescuing type 1 diabetes, right, with a beta cell implant? Without the influence of functioning islet cells, what is missing there? What kinds of things might be missing?
Absolutely. So, I love the saying, the enemy of the good is perfect. And so, I think you can get, obviously you can get rescue of glucose control with beta cell alone. I think there's a really nice paper from Pedro Herrera's group that recently showed this in Nature Metabolism. But, I think if you really want to get tight control, if you really want to restore the system, you have to have, as Roger Unger and Mark Housing pointed out recently, I had come up with this word, paracrinology, with that Roger Unger had already come up with. But if you don't restore the paracrinology of the islet, you don't really get that tight glucose control that you're expecting.
Yeah, that's a great comment, and also a great point. I wondered also, we talked a little bit offline about this sort of, again, I've said this a few times recently, the primary cilia and beta cells is having a moment.
Yeah.
How does that relate to the landscape you're talking about with the beta and alpha cells in this paracrine, environment?
Yeah, I think that's a really interesting idea. If the alpha cell and the beta cell are communicating, having that organized into microdomains makes a lot of sense. And you can also imagine that being a target in diabetes of how communication might be lost without just having massive changes to either cell. And so I think that's a wonderful idea. I think we're very interested in understanding from the alpha cells perspective, what are the components of the alpha cell cilia, but then also what are the components of the beta cell cilia that might be sensing amino acid or alpha cell tone. And I think that there's going to be some really cool work coming out of that.
Yeah, no, it's really coming out like a flood too now. So it's a really exciting times. Let me ask you, with it, if you can comment just a little bit on the alpha cell cilia,
Mhm.
where is that playing a role, just sort of high level?
Well, so I mean, we have not done exhaustive. We have done a little bit of studying of the alpha cell cilia so far. I would expect that there might be some of these amino acid transporters and things like that enriched, as well as GPCRs that we know, like somatostatin receptors, that are really going to be the first line of sensors to control the alpha cells. But I think that there's real opportunity for people. I don't think that's been studied as much as it should have been studied so far.
No, so here's a perfect opportunity for a student or a graduate student or postdoc to come and work with you on this project. But I
Yeah.
also feel like there's, one thing that's interesting is at the onset of type 1 diabetes, they've noticed, it's been noticed that there's a shortening of the primary cilia and beta cells in humans. And you wonder, I mean, has there been any data to suggest that there are changes in the alpha cell cilia at
You know,
onset of type 1 diabetes?
I am not familiar with that. I don't know if anyone's actually studied that. Yeah,
Okay,
I'm not sure.
more work to be done.
Yeah, I think that's a question for Jean Hughes, maybe.
Yes, no, I'm
Um,
sure she's
Matt.
looking at it. Yeah,
And Matt Marins. But
yes.
also other people, I think, Jacob Knudsen is, would be another good person who's studying the alpha cell to talk to.
All right, well, we'll get him on the roster, but thank you again for this really interesting talk and a great a great effort with your big lab and all the fantastic kind of unearthing of what's happening with the alpha cell in context of diabetes that you're doing. Thanks again.
Thank you so much. I appreciate the offer to come and tell you about our work.
It's great. We appreciate it too. Have a great rest of your day. Bye-bye.
You too. Bye-bye.